Next, science and treatment for rare diseases. And for a final break of the event, we hope you all had a great break and have enjoyed these last couple of days of the Global Genes Summit. My name is Miguel, and I'll be your host for today's session on advances in rare immunological diseases. This track is being live streamed, and I'd like to welcome our remote attendees and remind everyone that you can use our live Q&A feature. As of today, there have been many advances with the potential for cures in these types of diseases. Today with us to share their expertise and knowledge on these advancements is Manish Butte, Division Chief of Immunology, Allergy, and Rheumatology at UCLA, and Claudia Gonzaga-Jauregui, Senior Manager for Translational Genetics at Regeneron Pharmaceuticals. Thank you.
Okay, let's see. I may not need this if I have this. Can everyone hear me okay? Okay, Okay, that's us, and that's the laser. Okay, good. Thank you all for inviting me to come, and I'm happy to share what we've been working on in terms of advances for rare immune diseases over the next 2 hours, I think they gave me to talk. No, I'm kidding. Anyway, let's just get started. And as I mentioned, I'm at UCLA. My email address will be at the end. So if you guys have questions, if I tend to go fast, I'm an East Coaster, so I tend to talk very fast. But if you need me to answer some questions or whatever, just feel free to email me, and we can connect. So I don't have any relevant disclosures, and I'm not going to be talking about anything interesting from the point of view of disclosures. So I think Claudia has a slide like this, and we all do, because the cost of genome sequencing has come so far down at this point that it's become a mandatory for virtually every interesting rare disease. At this point, genome sequencing has come well below $10,000 into the few $100 range. We now do research-based genome sequencing for about $650 at UCLA. And it brings the price of genome sequencing is coming down further and further and further to the point where exome sequencing doesn't have anywhere to go. And so we're moving rapidly towards genome sequencing. One of the take-home points of the first part of my talk will be how important genome sequencing can be for identifying rare diseases. It can go even lower. This is actually a sequencing platform right here called the Mini-Ion. This thing can do a terabyte of sequencing. It's kind of incredible. It's for about $90 to do a terabyte of sequencing. The quality is not yet at clinical grade. We won't use this in the clinic. But this was used in West Africa during the Ebola outbreak to sequence pathogenic strains of the Ebola virus as it mutated, and presents a lot of opportunity for what we can imagine a few years from now being able to really do sequencing at the point of care. Just to remind everyone who's watching and in the audience, when we talk about sequencing, the genomic DNA is comprised of exons that get converted through RNA into messenger RNA, and then they code for a protein. Some of those proteins in the immune system can include things like antibodies, the antibody chains, interferons. These are signaling molecules that are used to resist viral infections, or cell surface proteins like the T cell receptor that are used to drive signals into cells. So the coding parts are the exons, they're colored here. The parts in between exons are called introns. And the vast majority of our DNA is either introns or DNA between the introns. In fact, when we talk about exome and genome sequencing, I like this slide that came from 23andMe, where you can see the whole genome comprises, if you take both strands, about 6 billion bases. Exome sequencing takes up about 1% to 2% of that, a portion of it. That's the relevant portion, the portion that codes for all those exons I just talked about. And if you're thinking about things like 23andMe or Ancestry.com, they're sequencing a tiny fraction of the exome or of the genome that comprises about a million bases, 0.03%. So it's really important to remember that 23andMe is for entertainment. It's not for making diagnoses of rare diseases. It's good entertainment, especially if you are interested in where your people come from. But in general, we don't like to recommend patients to waste their time and money. And think about 23andMe, unless it's Mother's Day, it's $99 on Mother's Day, but otherwise it's about $200. And remember what I told you, genomes are $600 at this point. So would you rather get all of this or just that for your couple $100? The problem with whole genome sequencing, I think Claudia will talk about this too when I looked at her slides, is how do you interpret all the parts that are between the exons? If I go back here to the previous slide, if you imagine a mutation or a pathogenic variant in an exon, it's going to have an effect on the protein, right? That's how we can say this protein is broken, and if we work our way back to the exon, or if you did exome sequencing, you'll find the variants of interest in the exons. This doesn't signal properly because the purple part isn't made properly. You go back to the exon to look for where the purple part was encoded in the genome. Does that make sense, everyone? So the introns, on the other hand, don't play any representative role inside the final structure of a protein. And so how do you interpret potentially pathogenic variants inside of introns? That has been an interest, especially if we're going to use the power of whole genome sequencing, we need to be able to interpret all of this green stuff that isn't the orange stuff. And so the approach that we've taken at UCLA, this slide got a little bit messed up here, is RNA sequencing in combination with genome sequencing. And so just to give you another example of exons and then coding sequences here in yellow and red and green, this particular gene, it gets spliced together to form this RNA and gets turned into the protein. And the protein, say, needs this red domain. That's the enzymatic power. That's the part that chews up some other thing and makes your engine go. Now, say there were a pathogenic variant in this intron, okay? And you didn't know that, and you wanted to know how that would affect things. Say the variant is here. What it could do is affect the splicing pattern so that this red exon drops out. And you can notice in the mRNA and in the protein, now it's missing the key part, okay? So if this protein were an engine and you took the engine out, now the car isn't gonna go. Does that make sense to everyone? So sometimes just cutting one little wire can take the whole engine will fall out. That's the kind of idea that we can think about when you're looking at interpreting intronic variants and their impact on the protein. Sometimes it's big enough that we can see an impact on the splicing, and we capture this information here then by RNA sequencing. So RNA sequencing is a complementary approach to genome sequencing. It allows you to interpret the intronic variants that lie in between the exons and may affect then the splicing of the isoform that's biologically relevant. So we now recommend at UCLA to combine genome sequencing with RNA sequencing. And RNA sequencing has to be done from the tissue of interest. For an immunologist like me, that means blood, or typically T cells or B cells, something interesting, sometimes bone marrow. These are the tissues where you're going to get the kind of information that's of relevance for looking at splicing and et cetera. Other doctors, other scientists may be able to use fibroblasts to do RNA sequencing from. They may be able to use other tissues, bone, et cetera. But in general, if you can get tissue of interest and do an RNA sequencing combined with genome sequencing, we have a paper that's part of our Undiagnosed Disease Network group that I'm a member of that has now shown that we can increase the diagnostic utility of genome plus RNA-seq to beyond exome and beyond genome alone. So this is going to become the new standard, especially again as prices drop, to get more bang for your buck out of all the genome sequencing that we're going to be doing. Okay, so genome sequencing, of course, sequencing in general should be the standard of care for all rare diseases. I think at a place like this, everyone understands that. It is still something we struggle with every single day. I spend hours per week fighting with insurance companies to get them to pay for exome sequencing, genome sequencing, et cetera. This is absolutely not a solved problem today. It is a major struggle for our patients. And we get letters like this all the time. This is an actual letter from one of my patients saying, we will not cover the exome sequencing because it is experimental or investigational. This is a very common thing to get. I got one of these just yesterday. I could have copied and pasted that PDF in. This is another one that I share. But for the doctors in the audience, many of you are nodding your heads because it is a major struggle we face. And so the second take-home message out there is, please, those of you who are advocates and who have the ears of payers, please fight for this. This is something that we aren't winning. We're not winning at all. We are constantly being pushed back by payers more and more. In fact, it's much worse in 2019 than it was in 2017. And maybe some of you will acknowledge that in your own practices too. We are having a very hard time getting exome sequencing done, to the point where we now have to do research sequencing because we just can't waste our time fighting with insurance companies. Blue Cross Blue Shield is particularly horrible at this. They set up a variety of barriers at some point, including AIM and these other sort of enemies of the people who eventually just wear you down through phone call after phone call, and We just move those patients to research sequencing. It is cheap enough that we can do research sequencing for our patients, but it's not the right thing. We really should be able to get clinical sequencing for patients. Okay, so in general, when you have a pathogenic variant, when I first learned about pathogenic variants and thinking about genomics as a way of solving problems, I used to think that when you make a mutation, it's going to break something. So if you have a fish hook and you have a mutation, a pathogenic variant, it's going to be a loss of function. The hook doesn't work as well, you're not going to catch as many fish. But believe it or not, there are lots and lots of pathogenic variants out there that actually are gain of function variants. And in immunology, we have about 20 diseases at this point, 20 genes, where we know pathogenic variants arise. And the biological phenotypes for these patients can be everything from infection, allergy, malignancy, severe autoimmunity, or even autoinflammation. So the idea behind a gain of function mutation is that there's a pathogenic variant that leads to an acceleration of some enzymatic process, and it leads to an immunological loss of function, but an enzymatic gain of function, or if that makes sense. Okay, so if the next part of the talk is how do we treat that? So one idea, if you just think about signals and receptors and driving sort of signaling pathways, and then eventually turning on genes as a very schematized view for how signaling processes work, then in a gain of function, you may end up with some kind of gain of function could drive more signal. Wow, what happened there? And turn on more genes. And instead, what we'd like to be able to do is use inhibitors, biological inhibitors, to block these pathways and slow down gain of function diseases. So does that make sense to everyone? If your biology is pressing the accelerator, then we can use a biological inhibitor to press the brake. Now it's important not to press the brake too hard. Many of these drugs were designed to press the brake all the way. For example, in cancer, if you want to shut down some pathway in order to choke off some cancer cells that need this particular signaling pathway as part of their biology, well, that's great. Pour on the drug. But in gain-of-function immunological diseases, we want to be able to tune their biology down to wild-type function, if that makes sense. So it's really important to be able to monitor the signals as we give them the drug. And we want to slow them down, maybe not to a complete stop, but down to biologically normal levels. That's part of the challenge that we face when we use drugs off-label to treat these diseases, but it's something that many of us have become good at, and it's part of our standard of care. And I'll give you an example. So this is a 28-year-old who had a lifelong history of thrush because she has candidal infection in her mouth. And then she also has fungal infections on her nails. She was sequenced in a bygone era, and somebody thought that she had a leaky version of SCID. In fact, what she has is a pathogenic variant in one of these gain-of-function diseases in a gene called STAT1. STAT1 lies downstream of many of the interferon receptors, like interferon gamma. And this has now become a very well-described disease. There are probably over 300 patients in the world with gain-of-function mutations in STAT1. Is that rare in this crowd? I think it's pretty rare. Gain-of-function STAT1 can cause infectious complications in virtually every tissue of the body, and candidiasis is a major one. Virtually all the patients have thrush. But they can have infections, pathogenic infections, including atypical mycobacteria and bacteria and viruses and skin infections, et cetera. They also have a lot of autoimmunity, and that autoimmunity can include everything from organs like the liver and thyroid, and cytopenias as well. So patients with gain-of-function STAT1 can look like CVID patients. They can look like SCID kind of patients with leaky SCIDs. They can look like patients who have some hypogammaglobulinemia. They can look like patients who have a combined immune deficiency. The phenotype is extremely broad. This disease taught me an important lesson: to move away from the concept of one gene, one disease. We really don't believe in one gene, one disease anymore in immunology because of genes like this, where the phenotype is so broad. And so this disease is highly amenable to blockade of the receptor pathway because these STATs lie downstream of many cytokine receptors. And so if you can block either the cytokine receptor or the JAK kinase that phosphorylates and activates the STATs, then you can slow down this disease, just as I showed you with the slides. Now, there are many cytokine receptors and many JAKs and many STATs, and so you have to be able to pick the right drug to target these right pathways in each patient, and that is not a solved problem. It's still actually quite empirical at this point, but there are lots of hints that we have in immunology to know which inhibitor to choose based on which pathways are deranged. This is totally screwed up. I promise you I previewed my slides before this talk. But, so there are 3 drugs that are used now in the literature, tofacitinib, ruxolitinib, and baricitinib, that are FDA approved. They're made for cancer. We use them for this disease because they block the relevant JAK pathways. I'm going to give you a second case. This is a boy who came to our hospital. He had these giant lumps on his forehead and also on the back of his neck. We did a biopsy of one of them and found a spherule. A spherule is a fungus. This is a very obvious diagnosis to a pathologist. This is called coccidioidomycosis, or valley fever. It's a pathogen that lives in the soils of California, Arizona, and some parts of West Texas, and it goes all the way down to the tip of Argentina, where this disease was first identified in a Uruguayan soldier in the 1890s. As the soils dry out, or agricultural work of the soil, or as there's construction, the spores are released, they go into your lungs, and in about half the people, they do nothing. They're asymptomatic. But in about a third of the patients, they get what's called valley fever, which is a lung infection that's a pneumonia. And many of these patients do fine with antifungals and are able to be treated. And about 1%, the rare side of things, the fungus spreads beyond the lungs and into all the tissues of the body, the bones, the joints, the brain, the meninges, et cetera. And these are the very, very sick patients. It's called disseminated coccidioidomycosis. And so far, it's been described with certain states like immunosuppression, for example, for transplant or cancer and HIV, or in pregnancy, to be highly susceptible states. But if you take the people who aren't pregnant, our 4-year-old patient was not pregnant, or immunosuppressed, and he didn't have HIV, then you fall now squarely into the world of rare disease. And we wanted to try to understand, did he have a rare disease as the cause of his susceptibility to this infection? It has been described in the past 8 cases in the literature where rare diseases of the immunological pathways of Th1 immunity, type 1 immunity, including interferon gamma and IL-12 receptor, as being susceptibilities. Now, these are defective type 1 immunity as being a susceptibility state. But historically, even through the 80s, people realized that excess of type 2 immunity was also a susceptibility state. So this is not a defect of type 1 immunity, but a gain in a way, a biological gain of function in type 2 immunity. And the evidence for it has been in the era where people didn't know what T cells from a hole in the ground, like in 1982, they still knew that things like high levels of IgE or eosinophils or total IgE were actually highly correlated to patients who had disseminated disease and who would get much sicker than those who didn't. So historically, we used to think of these type 2 cytokines that drive type 2 immunity as being relevant to allergy. Okay, so when you see IgEs in a patient, you may say, oh, you're allergic to this, right? So that's the context that this was presented in through the 80s and 90s and even to today. People think that patients with lots of allergies have a susceptibility to coccidioidomycosis. But what I'm going to tell you is I think the balance between type 2 and type 1 immunity was staring them in the face, and it didn't occur that this is actually one playing off of another. Okay, so the cytokines IL-4 and IL-13 come from the fact that fungi have, as in their cell wall, chitin, and chitin drives innate immune cells in the pulmonary epithelium to produce key cytokines that then turn on T cells and all the innate immune cells to make and respond to that IL-4 and IL-13. So this is back to our child. He had a PET scan when he got admitted. This is last spring. He had lesions in his tibia, in his wrist, in his ribs, in his clavicle, in his sternum. This is his wrist here. You can see it was wider. This is the part that lit up on the bone scan. And a few days later, this grew out. This is a highly destructive fungus. It eats tissue, blood, bone, everything that it can. He needed many, many surgeries to remove these lesions as he was on triple antifungals, and we could not slow down this disease. He was heading for the door. These are the lesions on his scalp. This is on his clavicle. This is on his vertebrae. It was on his rib. So taking a hint from the previous publications, we looked at IL-12 signaling, we looked at interferon gamma signaling. We found that when we gave his blood cells IL-12 and then looked for phosphorylation of STAT4, which is the key STAT downstream of this JAK-STAT pathway of an IL-12 receptor, that unlike a control patient who produced lots of signaling in response to IL-12, in his case he just had a little wisp of signaling, a tiny amount of IL-12 signaling. So what's the relevance of that? Well, IL-12 is needed to make a type of helper T cell called Th1. type 1 immunity. And type 1 immunity is needed to make interferon gamma, and interferon gamma is needed to drive your macrophages, innate immune cells, et cetera, to fight fungi. So if he had poor IL-12 signaling, we guessed that he should have low numbers of Th1 cells. Now in children, and in you and me, there's a high amount of Th1 cells to Th2 cells. The ratio is something like 4 to 1, even all the way up to 8 to 1, depending on certain ages. And we said, let's test that. Does he have low Th1 cells? And the answer is yes. When we looked at his T cells, he had a control person has a ratio, you can see here, of 21% to 11% to 2 to 1 ratio here. And here he had just a tiny amount of Th1 cells. So biologically, his low IL-12 signaling meant he could not make type 1 immunity. So we gave him interferon gamma. It's a drug. We can use it off-label. We gave him this drug, and he started to get a little better. Some of his lesions, like this lesion on his scalp, went away, but unfortunately this one grew. So it wasn't perfect. We were able to slow down his disease a little bit, but it wasn't enough. So we looked back at our T helper diagram and we said, oh, look, he's making so many Th2 cells, and Th2 cells make IL-4, and IL-4 suppresses IL-12, and this little chain reaction of events is something we can exploit, because now there's a drug to block IL-4. And we said, if we could block his IL-4, and maybe we should be able to rescue even more of his Th1 function. And so that's what we did. We took a biological called dupilumab, which was invented for eczema, but we said, let's use it in this severe fungal disease. And lo and behold, in vitro, we could get him up to 30% Th1 cells. And so we started him on dupilumab, and we didn't know what dose to use, so we simply gave him enough until we could utterly suppress IL-4 signaling in his cells. At that point, we said, well, he's got no more IL-4 function, he should be able to get some benefit from this. And indeed, he got a huge benefit from it. His Th2 cells went down, and his Th1 cells went up to the ratio of about 1 to 1, and his lesions completely melted away. His spinal lesion went away. I didn't show you that. All his scalp lesions went away. The one on his scalp, this is just after he started dupilumab, to about 8 weeks later, completely melted away. Okay, so repurposing drugs like interferon gamma and dupilumab is essential for taking care, and ruxolitinib, and all the drugs that I mentioned earlier. This is essential now in the advanced era of immunology. We need to be able to identify pathways that have gone awry. We need to target those pathways using other people's drugs and use them for our kids and be able to save their lives. That's key to the way we work now. And you need to be able to monitor these pathways using immunological testing. Research and patient care are the same in rare diseases. I don't have to tell that to you guys, but that's the way we have to manage patients like this. However, we get pushback about this idea called evidence-based medicine. I had a long phone call with some person at Blue Cross Blue Shield who said, why are you doing this? Show me a paper where this kind of thing works. And I said, there are only a few patients in the world like this. I can't do a controlled trial. Do you want to call this mom and say, you get to be placebo? Do you want to do that? Tell this mom that her kid gets to be on placebo and there's only 2 patients in this study? That's not going to happen for very rare diseases. We have to switch our mentality in rare disease care from evidence-based medicine to what I call rational-based medicine. You see a pathway, you apply due diligence like hell, and if you can see that this pathway is the right pathway to go after it. Try it in the lab, test it, we did that. And then go after it in the person and see if you can make a difference. There's no other way to move forward in rare disease care unless you can take this kind of step forward. You have to have the laboratory support, you have to have that kind of backing. But we get a lot of pushback because there's also some dollar signs there. By the way, in our patient who we're trying to get ruxolitinib for, this rejection came through a couple days ago. Medicare does not allow for drugs to be used, even if they're described in the peer-reviewed literature. So Medicare needs to do better. It's not just Blue Cross Blue Shield, it's Medicare too. It's everyone. They're all out to get our patients, but we have to persevere. And when you start talking about using personalized medicines for individual patients, repurposing drugs that are extremely expensive, somebody has to bear this cost. And I don't really have an answer for this, except I know the literature does not help here. Leadiant, who is an incredibly innovative company, has this particular baggage around their neck. They bought this rare disease drug called CDCA. It's deoxycholic acid. It's used for a very rare xanthomatosis disease. The price of the drug was $300 per patient per year. They raised it to $153,000 per patient per year. They took advantage of an opportunity of a small clinical trial. This has been done for colchicine. Colchicine, many of you know, has been a drug that's been used by the Egyptians since 2,500 years ago. It is now, of course, FDA approved only by by one vendor and has become an extraordinarily expensive drug. And so we have to be aware that this is going to happen to us. We have to fight these battles, and it's a losing battle right now, but it's something we have to do if we're going to make advances in immunology care. So thank you. Here's my tiny 9-point font email address. I don't know what happened to the fonts, and thank you. I think we're going to take questions afterwards, and here's Claudia.
Great, thank you. Yes, so there will be some overlapping themes, because the thing, as Manish said, is there's a lot of development on genomic sequencing and its application in rare diseases, but hopefully a lot of complementary stuff. So the main message that I want to bring today is how achieving an accurate diagnosis early on can help to identify effective therapies, in this case for rare immune disorders, but more in general for rare diseases. So I want to start just saying, like, the human body is really an amazing machine, right? So you have a machine that it's able to respond to the environment, and in the case of the immune system, to respond to a variety of pathogens and exposures and viruses and fungi and all different types of bacteria, etc. But that response is really driven by the genetic makeup of the person. So in order to really understand how a person interacts with their environment, and in the case of immune disorders, how these may be dysregulated, we need to really also understand what's the encoded variation that that person has and how that determines that response. So really, the path that to enable genomic medicine or precision medicine and the state at which we are now has been long, and it has started with basically understanding human genetic variation. So we started, as Manish pointed out, with the Human Genome Project, which was really costly, but it gave us the basic layout of the human genome, what's the reference sequence, and what it's in there, what's the template. Later on, we had the personal genomes sort of boom, where we started to see and trying to better understand how different people differed from each other, how similar we were, but also what were the differences that determine each person's individuality. And then we moved into identifying individual variation that can be used to determine the molecular cause of a particular disease, and how to use that information to guide management and treatment for rare disorders, and also applying that knowledge of that variation to identify new molecular diagnoses for rare and undiagnosed disorders. So this is the same graph. So I won't go into the cause, but as you can see, it has decreased dramatically from the original Human Genome Project. And then, as I mentioned, a lot of that was driven by development of technology for sequencing. Then we had the personal genomics era, but then it has just been about a decade since we started really using genomics for clinical diagnosis, and this boom of applying genomics for disease diagnosis and novel gene discovery. So this is in 2010, where we published the first proof of concept that you can identify from genomic sequencing data the individual genetic cause of a particular disease where you don't know what the gene is. And so from there, as Manish mentioned, so we have different approaches. We have whole genome sequencing, but then we also have exome sequencing, which interrogates 1% of the human genome, since we have two copies, one from mom, one from dad. So if you sequence all the exome, that's about 2% of the genetic information that everyone has. And that's the part that codes for the proteins, and those are the parts that perform the major biological functions. So you can see there from the cartoon, and very similar to what Manish showed, the the exons and the introns of the gene, and that's just that little sliver of the genome. And this is what the data actually looks like. So you have, from exome sequencing, you have these peaks where you are just sequencing the blue parts here, which are the exons, versus with whole genome sequencing, you have coverage across all the regions, including the introns and the regions in between. However, this also comes with some differences. So there's a lot of debate, and I think, as Manish mentioned, as the cost continues to go down, we'll see more of a transition to whole genome sequencing, complemented by other technologies such as RNA sequencing. But still, there's a lot of utility to exome sequencing. And I think, in general, it's a bit more accepted by insurance to be paid rather than whole genome sequencing. So again, some of the differences, whole genome is still costly, a bit more than exome. So at the time where we did this, exome was about a third of whole genome sequencing. That gap has been reduced, but there's still some difference. Also, generally, exome sequencing is performed at much higher coverage, so you have a deeper sampling of those regions, the 2% that you are actually sequencing, versus whole genome sequencing, because you're doing more sequencing across all the DNA, then you have to sacrifice that for less coverage in general. So that's another issue to consider. The exome and genome coverage is not exactly the same. There are also technical differences. So as some of you might have seen from the Rady's hospital yesterday, so they are doing rapid whole genome sequencing for diagnosis in the ICU and the NICU. And that's possible because whole genome sequencing has no capture steps, so you can just put all the DNA and sequence, and that makes it a little bit faster than exome. Exome has a capture step, and that introduces also a bit of a technical bias in terms of what the design is, what you call genes, and what you are sequencing in the end. Also, as I mentioned, for exome sequencing, you're focusing on a small percentage of the genome, and that's limited to the coding and variation of the genome that encodes the proteins, so you end up having about 10,000 or so variants, versus if you do whole genome sequencing, you are faced with a lot more information, and the question is still how much of that can we actually interpret. You have the possibility to detect other type of variation that you might not be picking up from exome sequencing, such as copy number variation and trinucleotide repeats or other type of structural variants that you will not be picking up from exome sequencing, and also potentially having non-coding variation that might be of relevance. For example, deep intronic variants that might affect splicing, as Manish explained. And, but really, in terms of the application for disease specifically, exome is still pretty good for highly penetrant Mendelian rare genetic disorders, because most of them will be caused by variants. As Manish said, some of them loss of function, some of them gain of function, but that are changing the proteins, and therefore you are likely to pick those up from exome, whereas whole genome sequencing might be more useful for some other disorders that are later onset, or they are not as severe, that they might have other genetic causes due to non-coding variation or copy number variants or other type of variation. So genomic sequencing has really made a huge impact into the discovery of novel disease genes and the study of Mendelian disorders. So there was a study before, but actually this one was published about a week or two ago, which I was really happy because this gives us updated numbers as to what the impact has been for gene discovery using next generation sequencing in blue versus sort of conventional methods here in orange from the 1980s up to 2017 here. And as you can see, the contribution to novel gene discovery has been really fueled by the application of genomic sequencing, because before you relied in these large pedigrees, these very big families segregating these diseases, but for cases where you had one patient with a rare disorder, it was really difficult to try to do candidate gene approach or linkage or some of the other more conventional genetic methods. Now I know that, and I keep hearing it here, the 7,000 number that a lot of people say there are 7,000 rare disorders, but really, and I think for many people that's the experience, a lot of the disorders are, so that's based on OMIM, which is a database of Mendelian phenotypes and disorders. But the experience probably of many people here is that their disorder has never even been described in the database. This is the first time that when it's published or when you get a diagnosis, that someone saw a patient like that child. So we actually think that although the number that most people handle is 7,000 rare disorders, we're likely going to reach at least 10,000 disorders, because a lot of them are very rare, ultra rare, and we're just finding these one case or two cases that haven't been ever described before. So in the case of immune disorders, I think there's been a lot of advancement in how to treat immune disorders, and that has to do both with our understanding of immunity, but also how to identify the pathways, as Manish was saying, and how to find what is the best molecule or approach to target those disorders. So very broadly, the immune system can be divided into innate immunity, which is non-specific. It's a kind of one-size-fits-all. So it's the way for the human body to respond to any kind of pathogen that it comes in contact. And it's composed by interferons, cytokines, complement, natural killer cells that just have a first response to this exposure and start sort of dealing with this infection or these strange thing that they are encountering. And then after that, you have adaptive immunity, which is much more tailored to the specific pathogen. It takes longer, but it is very specific. It includes the development of B and T cells and antibodies against the particular pathogen or molecule or protein that the body encounters. So understanding all these pathways really is important in how we treat immune disorders, because then we have disorders that are more of the innate immune system, that it's just hyperactivation of some of the innate immunity versus sort of defects of adaptive immunity. So I'm just going to give a couple of examples. So in the case of the innate immunity, for example, we have CAPS, so cryopyrin-associated periodic syndromes are a group of autosomal dominant autoinflammatory disorders that vary in severity, but they share features of rashes, fever, chills, fatigue. So in the first case for familial cold autoinflammatory syndrome, patients get into a room like this with air conditioning, and I'm wearing a sweater and I'm okay, but in their case, they start developing like a really, really severe cold. They have this rash and this fever, and it's really uncomfortable, really life-limiting. So in 2001, Hoffman and his group identified that mutations in the NLRP3 gene, which is the parent domain containing 3 gene. So this is why the disorders are called cryopyrin-associated syndrome, because NLRP3 is known as cryopyrin. So mutations in this gene cause familial cold autoinflammatory syndrome and Muckle-Wells syndrome. And then later on, it was identified it was a bit more of the pathway biology and how these mutations that were initially identified in these families actually cause this disorder. And going back to Manish's talk, so this is a case of a gain of function in this gene, cryopyrin, that lead to, so cryopyrin is here, and when you have that mutation that causes cryopyrin to be hyperactive and downstream to cause the overproduction of interleukin-1 beta, which is an interleukin, an inflammatory cytokine that then produces all these response towards, in the presence of cold, but normally you would produce it towards a pathogen or an infection, but it's just an overactivation of innate immunity. Similarly, we have disorders of complement, where you have an activation of this cascade that is also part of the innate immunity. And actually, for complement, there's a big array of disorders that have been associated with complement dysregulation. And also, to what Manish said, like it goes beyond like one gene, one disease, but understanding what the mutation, the molecular causes are, can give you insights into what the pathway is and the presentation, even if it's broader. So moving away from a clinical only diagnosis and into the molecular and trying to find the therapy that works best for the molecular defect, it's where I think precision medicine will move to. So in this particular case, so CD46, it's a membrane glycoprotein that is a regulator of complement. It's a negative regulator of C3b and C4b, which are other complement molecules. And so, again, this is just a cascade of activation of different molecules that eventually lead to the accumulation of certain proteins in tissues. In the case when there is a pathogen, in the pathogen that then destroy that pathogen and then help the immune system to mount an adaptive response later on. But in the case of complement dysregulation disorders, these membrane attack complex that forms downstream of all this cascade actually attacks the tissues of the patients and cause them a lot of different autoimmune disorders. So previously, single mutations in CD46 had been linked to susceptibility to different autoimmune disorders, including atypical hemolytic uremic syndrome, lupus, systemic sclerosis, idiopathic recurrent miscarriages, etc. But then we have identified patients that have 2 mutations, one from mom, one from dad, and in those cases, they develop a disease that is very similar to immunodeficiency, combined variable immunodeficiency with low IgGs and T cells that are defective in their activation. So in many cases, if we know that a given patient has a disorder of complement dysregulation, then we can potentially intervene at some point of this pathway, prevent the formation of the membrane attack complex downstream, and then avoid the tissue damage that causes downstream disease. So just in the last few minutes, I want to go over another disease that I've been working recently in the last 5 years or so, which is inflammatory bowel disease, mostly pediatric. And this is a disorder that's very interesting because it can impact pretty much all different types of the immune system. So inflammatory bowel disease, it's defined as a chronic relapsing inflammatory disorder of the gastrointestinal tract that presents in genetically susceptible individuals. And the most common form that most people know is the late onset, the adult onset, which is, has an onset of about 30 years old. And we have broadly divisions of Crohn's disease versus ulcerative disease and some IBD unspecified disorders. But they are mainly classified based on the portions and how spaced the damage to the gastrointestinal tract is. So in ulcerative colitis, you have mostly colonic involvement versus in Crohn's disease, you have patchy involvement of the tissue, but both of the colon and the ileum and the small intestine. So this disorder can be caused by defects in both innate immunity, adaptive immunity, gut epithelial barrier dysfunction, autophagy, so again, the full range of immune response. And we know that genetics plays a role. And so in the last 5 years, what we've been doing is studying pediatric cases of inflammatory bowel disease, mostly known as very early onset inflammatory bowel disease. And these constitute about 25% of the cases. And we have identified a lot of Mendelian genes that cause inflammatory bowel disease. In some cases, some of these are immunodeficiency genes that have gastrointestinal presentations, but there are still a lot of patients that remain undiagnosed. So this is just a list, and you can see that it's very big of the different genes that have been identified to cause Mendelian forms of inflammatory bowel disease. So more than 50 genes have been identified. But we can broadly divide them into these major functions that we're starting to see, which are immune dysregulation, epithelial barrier dysfunction, and environmental sensing. And those are kind of the main themes that are emerging as causing IBD. And this is important because you can treat certain types with certain agents or certain forms, whereas some of the others might be less tractable for treatment, and then you should focus a lot of the new therapy development to those subtypes. So in collaboration with a wide range of clinicians, both in the US and in other places, so mainly the International Early Onset Pediatric IBD Cohort Study, NEOPICS, but also investigators in other places, we have sequenced more than 3,000 patients with pediatric and very early onset IBD, both for diagnosis and for novel gene discovery. And this is a great example, I think, of how having a very rapid and effective diagnosis can enable appropriate patient management. So this is a kid that had inflammatory bowel disease. He had exome sequencing done. And at that point, so SIAP was known to cause immunodeficiency, but hadn't been linked to inflammatory bowel disease. And this was back in 2011. So they found a mutation in SIAP, which is the X-linked inhibitor of apoptosis. Based on this finding, they performed hematopoietic stem cell transplantation, and that basically was curative for this kid. And so as you see there, at 42 days post-transplant, the child was able to eat, drink, and there was no reoccurrence of gastrointestinal disease. So this is really important because we know for IBD, so there are certain types that, like in the case of SIAP, hematopoietic stem cell transplantation, it's actually curative. So you remove all the cells that are causing the immunodeficiency, you reconstitute the immune system, and that cures the patients. In other cases, we still don't have a therapy. And of course, We have new approaches now that we can start exploring, like gene therapy, which is delivery of the missing or the normal version of the gene into the specific cells using viral vectors that can rescue normal function in those cells, and also approaches like gene editing, where you can actually go and, depending on the type of mutation and the defect, either inactivate or correct the mutation in that gene and restore function. So this is just in general how I see the application of molecular diagnosis and genomic technologies in rare diseases for patient diagnosis, novel gene discovery, and therapeutic development. And with that, I'll end there. And thank you very much.
So if anybody has any questions, you can feel free to step up to the mic.
Thank you both for a great presentation. My name is Mark, and I'm with the International Pemphigus and Pemphigoid Foundation. My question is regarding therapy for regulatory T cells. It's kind of a cutting-edge therapy. I just wanted to see what your feedback and comments were on that type of therapy as the field moves forward.
Yeah, I can comment on that. There are a couple strategies that we can talk about even right now. For example, there's a small molecule called sirolimus that's a blocker of mTOR pathways. And as much as it is immunosuppressive in that way when used at high doses in organ transplants, when dosed appropriately, it actually promotes the development of regulatory T cells. And so in some autoimmune diseases, and I'm not sure pemphigus is one of them, by giving sirolimus as an immunosuppressant drug, you can actually boost regulatory T cells. Pemphigus may also have a humoral component, and so treating the humoral side as well as the cellular side of autoimmunity is important for autoimmune diseases that have both antibody-mediated and cell-mediated pathways of harm. Driving regulatory T cells may eventually lead to some improvement on the humoral side, but we have lots and lots of good strategies around blocking humoral autoimmunity, like rituximab and Velcade, and et cetera. Long term, there are a couple groups that have been working and have published papers on the idea of taking peripheral blood T cells and adding in FOXP3 into those cells. Those cells all are endowed with the key transcription factor of becoming a regulatory T cell, and they all become regulatory T cells. And the lifespan of those cells may be questionable if that's a durable therapy versus a short-term therapy. Those are things that can be debated. But at the same time, if you pour regulatory T cells into somebody's blood, we know this from mouse models, we know this is likely to work in humans too, that you may be able to simply drive up the number of regs and create antigen-specific tolerance across a variety of self-antigens. So that strategy is going to be used by two different groups, at least two different groups right now at Stanford and at UW, working on trying to endow FOXP3 into peripheral blood T cells. Longer term, I think the groups want to try to endow FOXP3 at a hematopoietic stem cell level in terms of fixing patients who have defects of FOXP3, like IPEX patients, not pemphigus patients. But in those cases, by fixing that, then they should be able to, under the endogenous promoter, make FOXP3 and thus make regulatory T cells. So there are many strategies here around different autoimmune diseases. Hopefully that answers your question to some degree. Silence. Don't be scared. Come on, ask a question. Go ahead.
My son has no thymus gland, so no T cells. And he was in the hospital last week and turned out he has Boca virus. The infectious disease doctors kept saying, oh, we just have to wait for the disease to run its course.
Yeah.
And I kept asking them, is that possible? Without any T cells, I mean, he gets his intro weekly, so that helps, but I was just curious because they used to think, oh, well, it should be possible. It should be able to run its course. I'm like, is it? And so that's just something that I was talking about last week with ID doctors.
So there are at least 3 different Boca viruses. Boca viruses typically cause gastroenteritis. Is that the phenotype that he has of Boca virus?
I'm not actually sure. They called me because they ran the Carius culture on him. and it came back negative initially.
Yeah.
And then a couple days later, came back with bocavirus. They didn't tell me which.
But
pathogen.
the symptoms he's having is
his
gastroenteritis.
symptoms were mainly a fever,
Oh, okay.
102 fever, crabby, a mild cough,
Yeah.
and then maybe he has astrovirus too. So he kind of goes through periods of loose or firmer stools, and so it's hard to know what's what there while we're waiting for his thymus transplant.
Yes. Right. So cellular immunity is, of course, important for fighting viral infections. I don't know if you want to answer. I can definitely jump in on this
go
one.
ahead.
Cellular immunity is important for fighting viral infections. We can't clear viral infections without T cells. And in general, humoral immunity is also important for luminal viral infections. We know patients who don't have antibodies, but who have plenty of T cells, have a very high susceptibility to enteral viral infections. So both cellular and humoral immunity are important for fighting gastroenteritis like bocavirus and also astrovirus, both GI infections. So how do you treat someone who doesn't have T cells until they get their thymus transplant? I presume there's some underlying known defect like FOXN1 or DeGeorge syndrome or TBX1 point mutation. Okay, great. Yeah, so DeGeorge syndrome. Yeah. There's good news that the thymic transplants are back online at Duke, and
Number 2 on the list right now.
okay, that's great. Yeah. So thymic transplants are used to regenerate the functional capability to make T cells. That whole process had been put on hold for a couple year and a half or so, because Duke was licensing out the technology to a company, which is part of the tech transfer process that universities go through, but it resulted in some delays, and it sounds like things are moving forward again, which is great news. One idea that some people use to treat intractable enteral viruses is to give immunoglobulin enterally. There are really sort of mixed and matched results on this. The idea is that polyclonal antibodies that I have, that you have, that you could be plasma donors for, if we've had those gastroenteritis and we've made protective neutralizing antibodies to them, then those antibodies will appear in the IVIG pool. And if you just pour IG instead of in the blood, but down the lumen of the gut, yes, some of it will be flushed down into the toilet, but some of it may also neutralize up some of those bacteria. We know in principle this works because this is the whole point of IgA, right? We make IgAs in our gut, in our luminal space, to grab those viruses and bacteria and flush them down the toilet so that they don't cause bad infections. Whether IgG can substitute for that for IgA because it's not as multivalent and doesn't interact with the mucosa in the same way, et cetera, et cetera. There are lots of questions there. But in intractable cases, there are many, many case reports now of people using enteral Ig as a strategy for fighting that. At the same time, giving IV Ig or subcutaneous Ig is certainly a good idea in patients who have defective T cell immunity, because the B cell immunity is going to suffer no matter what. So hopefully he's getting Ig replacement.
Yes, he gets his extra weekly.
One other thing is that there are some antibiotics that have been used for norovirus. Perhaps if you're familiar with those, but nitazoxanide, for example, is used to fight bacterial infections in the gut, but happens to have some activity against noroviruses and other enteral viruses. Nitazoxanide? I could come tell you about it later. It has worked surprisingly for viral infections too. In the back there.
Did you have a question back there?
Is that Ashni back there? Hey.
Your last slide referenced gene editing CRISPR technology. I want to know what your impressions are related to monogenetic rare diseases. Do you think that is a viable treatment option that we're going to see more of? And what are just your general impressions?
Yeah, so I think there's definitely potential, but I think we have to be very careful on how we move into that. So again, I think, and my fear with all the Chinese CRISPR babies is that will push sort of that field a little bit back, because I think in cases of rare disorders where there's no other therapy, where there's especially very early, you need to recover the pathway and neurodevelopmental or malformation syndromes, that could be a really good therapy to use if you have prenatal diagnosis or something. But I think because it has a lot of ethical implications, including germline transmission and that, so there needs to be a lot of careful thought and regulation in how that is applied, which is part of the reason why, like, for this case of this initial experiment that was done, I think there would have been much more gain from a much more compelling case of a disease where you can use this where you don't have any other option versus sort of editing for the HIV receptor.
But maybe it goes without saying, but this is a session about immune system, and the immune system is reborn from bone marrow and hematopoietic stem cells. And so germline CRISPR isn't necessary for immune diseases, that you can do hematopoietic CRISPR-Cas9 modification and then replenish the modified genome, hematopoietic stem cells, back into the patient and then reconstitute a normal immunity. That certainly has no ethical implications. That's the same kind of gene therapy we're using via viral delivery of transgenes, for example. CRISPR-Cas9 will just improve that process. There are limitations right now. Actually getting genes into hematopoietic stem cells is super not good right now. It is very, very hard to keep those cells stem and to actually make them last a long time. Unfortunately, delivering genes as part of CRISPR-Cas9 causes a lot of stem cells to not like to be stem anymore, and the efficiency is super low. So there are lots and lots of very, very important technical hurdles that have to be overcome, even to do it in hematopoietic stem cells. But those hurdles are being hit hard right now by many groups around the world, and there's no doubt that CRISPR-Cas9 will replace viral delivery of transgenic gene therapy in the future.
Great. Okay.
Wait, there's another question back there.
We were out of time, actually. I just apologize. But thank you, Manish and Claudia, for sharing your expertise and knowledge on this very important topic. This concludes our final break of the 2019 Rare Patient Advocacy Summit. We have a quick 15-minute break before moving into the main ballroom for our closing session. And would like to remind everyone that as we make this transition, that any questions that you may have for our speakers can be asked as we move rooms and on your way out. And check out our last resources in the back of the room. I want to thank you again for joining us in this last session, and we hope that you learned a lot and that you take the time to complete our quick survey on the mobile app. Thank you once again, and thank you to our speakers.